because we were just talking about the types of muscle fibers and the types of exercise that exist. What I want to talk about is how your skeletal muscle changes as you exercise. And this is a simplification, like you can take a class on this with it, which is exercise physiology. But the way that will help you understand it and have some predictive capacity is when you are using your skeletal muscles for exercise, what you're doing is you're communicating with them, okay? And what you are doing is, if, for instance, I do two different activities, and I'm realizing that there's a lot of in-between activities. If, Beast Boy right here, if I try to pick up something super duper heavy that's very close to the limit of my my contraction capacity, and I try to pick it up over and over again, what I am doing, and I can only do it a few times, by the way. If something is at 75% of your maximum capacity or more, you can't do 30 reps. There's like no way you can. This guy tried to lift up something that was almost at the limit of his capacity, and he may have gotten 8 reps out of it. What you're doing is you're communicating to your skeletal muscles, and what you are telling them is that I need you to be stronger. I need you to have greater sarcomere contraction capacity in the future, right? Does that make sense? You're not necessarily telling them that they're aerobically incapable, because they did, right? They worked fine as far as the aerobic needs. So when you do certain types of activity, you are communicating. If, however, what you are doing is, instead of lifting up something heavy, almost the limit of your capacity, you're lifting or moving muscles at far less than their capacity for long periods of time, what you're doing is you're saying, you're strong enough, but I need you to have a lot of longevity, right? And your muscles respond differently to those types of communication. So let's talk about how you can cause this to occur. That doesn't happen on accident. Okay, so what does not occur is that if I start using my skeletal muscles, I don't get any more skeletal muscle fibers to speak of, with an asterisk, because there are a few stem cells, so you can get a few. But during embryonic development, during the differentiation phase, what actually happens is you start out with little uninucleated skeletal muscle cells called myoblasts, but during differentiation, they actually fuse. So that you get a multinucleated skeletal muscle fiber, right? That fusion is like this incredibly hard word to spell called syncytium. And after syncytium, you can't go through cell division. Just think about trying to do that. Think about trying to make a cell that is a foot long and has 20 nuclei go through proper cell division. It just doesn't occur. So you are born with just about all of the skeletal muscle fibers you are ever going to have, okay? So you don't get really any new ones. So at birth, you've pretty much got all of them, with the exception of a couple of stem cells. So what happens after you hit adulthood? You know that you can change your muscles in adulthood. You know that you can. So what happens is, it depends on what type of activity you're doing. If you want your muscles to hypertrophy, which is the fancy word for muscles getting bigger and more massive, okay, what you are going to have to do is communicate to them that they are not strong enough for the activity that you are trying to accomplish. And you do that by contracting the muscle to about 75% of its maximum tension or more. If you were an elite athlete, they can calculate 75% maximum tension for you. But if you're you just do it based on how many reps you can do, right? If you can't do very many reps, you're probably at a high enough weight, right? So if you can do 8 reps or fewer, you're probably in this range, unless you're just being lazy, right? You do 8 reps, that's all I can do. This is called strength training. It's high intensity and it's low rep. And so what is going to happen is, if you do that over and over again, you communicate to your muscles that they're not strong enough, and they will respond. If you rest them and provide them with adequate amino acids and nutrient sources, what they will do is the anabolic process of protein synthesis to make more actin, more myosin, and more myofibrils. Does that make sense? So what's going to happen to the diameter of the muscle fiber if you do that? It's going to get bigger. The muscle fiber is going to get bigger, right? And this is, of course, preferentially going to train your white fibers, your fast lipolytic fibers, right? Those are the ones that you are using when you're picking up heavy stuff and putting it down again. So mostly white fibers that enlarge. Comparatively, those fibers have relatively little myoglobin. They make ATP primarily anaerobically, but they're very large and very strong. That guy in the picture is not going to win any marathons, right? But he is exactly who you want if there's a Volkswagen on your baby. That's the dude you want, okay? So with this, only a few contractions at a time are either necessary or even possible. And then this is the time that your trainer may have been right. Like, just like your mom, sometimes they get it right. This one, so if you really want to increase your muscle mass, you're trying to cause muscle hypertrophy, right? You actually want bigger deltoids and you want bigger pectoralis major muscles. What you're going to do is you're going to do this type of weight-bearing activity, high weight, low rep, but then you need to rest the muscle, because if you use the same muscle group in a similar way the next day, instead of using the available nutrients for anabolic activity, building muscle mass, you are going to be using the nutrients available for catabolic activity, which is muscle contraction. So resting muscle groups in between really using them for high weight, low rep is how you increase muscle mass. What happens if you're starving yourself at the same time that you're trying to increase muscle mass? You don't get any increase in muscle mass to speak of, right? Because these are energy and nutrient requiring activities to build this stuff. Okay, so what about the other type of activity? Realizing that there's an in-between. So this is lifting weights, and this is cycling, for instance, weak activity. So you are absolutely capable of pushing the pedals, right? And you can ride a bike for hours and hours. So this weak activity is far less than 75% maximum tension. Even if prolonged, you are not going to get a significant hypertrophy. The muscles are not going to get way more massive. And I'm not talking about sprinting cyclists. I'm just talking about somebody doing normal cycling. This long duration, low intensity exercise will communicate to your muscles that you are strong enough, but I need more longevity out of you. And in response, what they will do is the red fibers will preferentially be trained, and those make ATP primarily aerobically. And in order to support long-term activity, they will develop more capillaries, right? It will also develop more myoglobin and more mitochondria. That makes sense? In addition, during this process, you increase muscle tone without increasing muscle size. And as a matter of fact, even though you may actually gain some muscle mass, the muscle probably won't get bigger. So for instance, if you start working out and you're doing a lot of this activity, in addition to losing subcutaneous fat, because this is pretty good for losing subcutaneous fat, you can actually, for instance, tone the muscle so that even though you're slightly heavier, your genes fit better, right? Because the muscles are actually more toned, right? They kind of slim down, you're preferentially training the smaller fibers. Does that make sense? Okay, so this also is the type of activity that forces beneficial changes in the cardiovascular and respiratory system because you need them to support long-term activity in this muscle tissue. So what do you do if you want to, for instance, get the benefits of both? Well, you have to do both. It's two different kinds of training that you have to do. You want to increase your muscle mass because you either want bigger muscles or you also want the benefit of increasing muscle mass, which is an increase in metabolism. Then you have to do weight training. And if you want to slim down your muscles, tone, and make them available for long periods of time, then you have to do aerobic activity. So you're already behind everybody. Yeah. You're like, damn it. Okay. So what happens as you age? You know it's not good news. So as you age, generally you get lazier. And since muscle tissue is energetically expensive to maintain, if you don't use it, you start to not put as much energy into maintaining these anabolic processes of building muscle mass. So you start losing your muscle mass usually around your 30s, and you continue until you die losing your muscle mass. You decrease muscle mass between 30 and 40% total as you age, but the vast majority of that is just use it or lose it. You can prevent a lot of that loss just by staying active. You are less active in almost every single situation now than you were 10 years ago. There are probably a few people who are the exception to that, but most people get less active as each decade goes on. Part of that is that you don't have to wiggle as much. You don't enjoy it as much. There ain't nobody to play with, right? You're like, I'm going to go jump on a trampoline. You got too much crap to do. In addition, you're trying to get a job in which somebody else runs around and you tell them to run around, right? That's what you're trying to get. And you lose the benefit of actually all of the wiggling. So your fibers will decrease in diameter as you lose some of the myofibrils because you don't use them as much. So what happens if you ever damage your skeletal muscle? Well, skeletal muscle is amitotic. If you ever damaged an entire fiber, the fiber is gone. But you don't usually damage entire fibers because they can be up to a foot long and they're multinucleated. What generally happens is that you damage a part of a fiber and then the two ends will still stay alive. So for instance, let's say you had an emergency abdominal surgery, like an emergency C-section, like I got into a car accident and the baby was about to die and I was about to die, right? Then they're going to get in there as quickly as they can, right? And they can even do this slice, although they usually do this slice. Let's say that they did that and you've got your transversus abdominis. Your transversus abdominis is your packing tape muscle that holds your guts up here. And so with the transversus abdominis, if you slice it this way, what happens is you don't lose the cells. But what happens is you lost your attachment point. So instead of having an attachment point that goes like this, and you got packing tape that keeps everything nice and flat, you got two of them, right? You get a little pooch in the middle, right? However, if you instead, when you do a planned C-section, they try not to even cut the fibers. They like separate the fibers and slice between them. Is what they do. Because they know you'll lose some tensile strength by actually doing that. So under what circumstances do you ever really damage an entire skeletal muscle fiber? Like a necrosis kind of thing, like a brown recluse bite or something that causes tissue necrosis or snake bite? That can kill off whole cells. And you don't get them back. You can have like a softball-sized hole out of the muscle tissue. You don't get the tissues back. Okay, so satellite cells, by the way, are just these cells that have their muscle stem cells. They can make a few. Okay, so now let's just briefly talk about smooth muscle tissue, how it's different, how it's the same from skeletal muscle. We don't talk about cardiac muscle in this chapter because we're going to do it later. Cardiac muscle will be super duper similar to skeletal muscle. So smooth muscle is called smooth because it's non-striated. It's also under involuntary control. The next set of notes we'll do is autonomic nervous system, and the autonomic nervous system controls smooth muscle. The cells, you would think that they might not contain actin and myosin because they are not striated. They do, but the actin and myosin doesn't line up, so you don't get any stripes. So look, if you blow up one of those, you'll see a thin filament and a thick filament. That's myosin and that's actin. But they don't get striped because these are all going at different angles. Do they still do cross-bridge cycling? Yeah, they still do cross-bridge cycling. They have the same kind of thing going on. But they don't end up with the stripes. In addition, they don't just run this direction. They run from a bunch of different directions. So when a smooth muscle cell contracts, it kind of wads up to the middle, which is okay because that's good for the wall of a hollow organ. Okay, so they do have actin and myosin. They're not controlled voluntarily. The cells are much smaller. They're spindle-shaped and they're uninucleated. They do maintain the capacity to divide into adulthood. So, for instance, that same emergency C-section. So if they had to go in an emergency C-section, they slice here. I could lose some tensile strength with my skeletal muscle, but my uterus, the smooth muscle in the wall of the uterus, has the capacity to do cell division still. So can I get most or all of those cells back? Yeah, potentially I can. I mean, there's no guarantee. So you're less likely to actually form scar tissue in the smooth muscle. Okay, the other thing is that the contractions are longer, but more slower, but more sustained. And that is because the cross-bridge cycling is really slow in smooth muscle compared to skeletal muscle, which is fine because it's not an emergency response tissue, right? And so slower cross-bridge cycling, it also means something about how quickly it uses up ATP. If you have slower cross-bridge cycling, you can make ATP last longer, right? So they're more sustained. In addition, the calcium pumps on the SR are kind of pokey. They just like pump pretty slowly, which means every time you release calcium, you get a long contraction before you actually pump it back in, right? So these are like the sloths of the muscle world, right? They're not in any particular hurry. Okay. The strength of contraction is similar, but you can maintain a sustained contraction. And then the other thing is smooth muscle, unlike skeletal muscle, works best when it's stretched. When smooth muscle is stretched massively, 2 to 8 times its original length, that's when it has best access to cross-bridge cycling. With skeletal muscle, it doesn't work very well from here, right? That's not, if you wanted to pick up something really heavy, you know that you don't start from here, but you also don't start from here, right? That makes sense? You start somewhere like a little further back, right? With a little bit of stretch gets you optimal length, but not 2 to 8 times its length. So when does your stomach contract best? Right after Thanksgiving dinner. That's when the smooth muscle in your stomach contracts best, right? When does your uterus contract best? Childbirth. That's when it contracts best, right? That makes sense? Okay. And then the other thing is that with skeletal muscle, under normal circumstances, the only reason a fiber contracts is because the nervous system tells it to. But with smooth muscle, there's multiple ways to start it. First off, it has spontaneous electrical activity. So it has a pacemaker activity, kind of like cardiac muscle. So what happens in smooth muscle is you have this pacemaker potential in which there are these cells on the surface of smooth muscle cells, kind of like cardiac muscle cells. And they are sodium and calcium channels. And they work kind of like leak channels. So as long as you have a dependable sodium and calcium concentration gradient and a bunch of leak channels, then what happens is it just leaks in. It's sort of a dependable rate, and then when it hits an action potential, hits its threshold, you get an action potential, right? And then the action potential is kind of long because, of course, the calcium pumps aren't in any hurry, right? Everything's just like farting around. And then you repolarize back down, and then you start the whole damn thing over again. So what I'm telling you is that smooth muscle doesn't need the nervous system to tell it what to do, although the nervous system can tell it what to do. The nervous system can tell it to speed up or to slow down. Right? It can say, I know that you were on your way to a contraction, but now I'm going to hit you with something that depolarizes and you'll get there faster. Or I know you were on your way to contraction, but now I'm going to hit you with a neurotransmitter that hyperpolarizes and you'll do it slower. Right? So the autonomic nervous system is actually what can slow down or speed up contraction with the nervous system. So unlike skeletal muscle, if you release acetylcholine, what does it do? Excitation or inhibition? Excitation always. With smooth muscle, you can either actively excite it or you can actively inhibit it. Okay? And that'll be important today. And then the other thing is that smooth muscle can also be motivated to contract by hormones. We didn't see that with skeletal muscle. But with smooth muscle, probably doesn't surprise you at all that like a hormone called oxytocin will cause uterine contraction, right? For that matter, hormones called progesterone and estrogen can cause uterine contraction every month, right? So hormonally, but then in addition, it's not just women because you can get super stressed out and release stress hormones and it can give you the squirts, right? Like I'm so damn stressed out, apparently I'm going to poop 5 times before I give this talk, right? Or you can be one of those people who get stopped up every damn time. You're like, I am not going to poop. I think it happens every time I go on vacation. I'm like, in Italy, everything's wonderful, but I literally think I'm going to croak because I haven't pooped in 5 days.
Okay, Melanie.
What is happening?
I'm just going to say, I hope it does that to you when you're in Italy because it does it to us every time we come to your class. It does not. We can 10,000 come to your class.
You poop? You're going to like nervous poopers during... Just have to talk about poop, and people will remember it. Okay. In addition, paracrine agents can cause smooth muscle contraction. When your stomach is full, it will release paracrine agents, which will cause it to contract. When your stomach is stretched, that alone will also make it contract. So there's a bunch of different factors. And then the last thing is just talking about the 2 different categories of smooth muscle that exist. So the type of smooth muscle that most people are talking about most of the time is the second kind, and it's called single unit smooth muscle. And let me explain to you why it's called that so that you can remember. Do you see that these cells are connected via gap junctions? Do you see that? They're connected via gap junctions. This, although it doesn't look like it, is what the axon terminals look like in the autonomic nervous system. So what's going to happen is as soon as I release this neurotransmitter that causes contraction, they're all going to contract as a single unit, hence single unit smooth muscle. That would be in the wall of the uterus and the wall of the digestive tract, right? So these all behave like a single unit, and they have gap junctions between them. And then the other ones are ones that you can control individually. So this is called multi-unit smooth muscle because they behave like they're multiple different units. Do you see no gap junctions in this figure? Right? No gap junctions. You have the capacity to communicate with each one. Okay, so with this, what you want is you want to be able to control the cells individually. You don't want them to all work like one big wave. Like for instance, the cells that are responsible for controlling the curvature of your eye for focusing or the diameter of your pupil, you can like make little bitty changes. They don't all do the same thing. The muscles that are responsible for the erector pili muscles, right? You can stand some of those up and not others. Also, the smooth muscle that's in the walls of your blood vessels. You need to be able to make little tweaks. So this is single unit smooth muscle and multi-unit smooth muscle. I just remember it because this one behaves as a single unit and this one does not. Okay, but last little tidbit is that, of course, if it's on the last page of your notes, what do you know? Yeah, and you're going to miss the questions unless you read the last. I always write relatively easy questions over the stuff that's on the last page of notes, but people are like, I haven't seen that in 6 weeks. I don't know what the hell you're doing. Okay. Okay, so now we are going to do autonomic nervous system. And autonomic nervous system is not hard and it's not long. So it is wordy, though. Okay, so let me introduce you to sort of what we're talking about. First off, how many of you guys remember enough anatomy to know what I mean when I say autonomic nervous system? When I say autonomic nervous system, do you remember whether we're talking about CNS or PNS? PNS, it's part of the peripheral nervous system, yes? And when I say autonomic nervous system, do you remember whether I'm talking about afferent or efferent function? It is, nope. Autonomic nervous system is efferent, right? So let's just run through this one time and I'll tell you where we're focusing today. This terminology will get you if you don't brush it off from anatomy, but you did learn it before, okay? So here's where we're not going to be today, but just to make it a complete story. We are not talking about sensation today. Sensation will gather information from sensory receptors in a bunch of different places, right? And then send it to the central nervous system via the afferent nervous system, right? Okay, now, what we are going to be focusing on is this region right here, in which the central nervous system sends information out to effectors. And we just, in the last set of notes, talked about the somatic nervous system, because the somatic nervous system has what for its effector? Skeletal muscle. Skeletal muscle is the effector for the somatic nervous system. We already know that. We know what neurotransmitter is used. We know how all of that works. So today what we're going to do is we're going to take the skeletal muscle, the somatic nervous system, and use it to compare and contrast against the autonomic nervous system. The autonomic nervous system is also sometimes called the visceral motor division. Visceral motor, because it innervates the motor activity of your guts. And it uses 3 tissues as effectors. What are the 3 tissues that the autonomic nervous system controls? Smooth muscle, glands, cardiac muscle. So we are going to smooth muscle, glands, and cardiac muscle. Okay? So that is the autonomic nervous system. And then, of course, today we will get into the sympathetic and the parasympathetic branches of the autonomic nervous system. So these concepts are not very difficult, but if you are having vocabulary problems, then you're going to need to make sure that you get over those vocabulary problems. That's why I try to teach this in detail in anatomy so that people will remember it in phys. Okay, so what we are going to do today is we are going to start from a place that we know, which is how skeletal muscle works, and then we're going to compare it to a little less familiar place. So there's a bunch of, before you begin, just talk briefly about what the nervous system does in general. The nervous system does 3 basic things. Information in, that's sensation or afferent activity. Information processed, which goes on in the central nervous system, the brain and the spinal cord. Okay, that's called integration. And then information out, which is motor or efferent activity. Does that sound familiar to you? Okay. So, see if I can make this question make sense to you. The last class didn't quite get it when I was asking it, so I have to figure out how to ask it. Okay, so anatomically, structurally, The brain and the spinal cord is where most of the neuron cell bodies are. That's where most of the decision makers are. Yes? Okay, so now anatomically what I want to know is what is out here? What nervous system structures are out here? Yeah, it is. Say it again. Yeah, it's primarily a big bundles of myelinated axons wrapped in connective tissue. What do you call a big bundle of myelinated axons wrapped in connective tissue? It's a nerve, exactly. So pretty much all the decision makers are here, and this is just wiring. Yeah, it's just wiring that's going to and from. Does that make sense to you? So you really don't make any decisions with the peripheral nervous system to speak of. I mean, there's always little exceptions because there's exceptions everywhere, but very few decisions. Okay, so now the afferent nervous system, just to make sure that you understand it, the afferent nervous system gathers information from sensory receptors. We have all different kinds of senses. We've got our special senses, the 5 vision, hearing, taste, smell, equilibrium. And then you have your general senses, touch, temperature, pressure, pain, all that kind of stuff. We're not going to be talking about that stuff today. So we are going to get to the efferent or the motor division. The efferent or motor division carries action potentials away from the central nervous system to effectors. Effectors, efferent. So the somatic nervous system, okay, so stick with me for a second. The somatic nervous system is the simplest. The somatic nervous system has one neuron, and the neuron cell body will be in the gray matter of either the brain or the spinal cord. And then what's going to happen is that neuron only sends its axon out to the skeletal muscle, meaning that there are not 5 synapses on the way. There is one, right? It's a very simple anatomical route. So what do I mean? I mean that the neuron that is controlling the muscular contraction in my little toe, yeah, the neuron that is controlling that is up here in the lumbar region of my spinal cord. That's where the neuron cell body is. And it is going to send that information down an axon that runs down my sciatic nerve and then branches off and goes to my little toe. And I form one synapse in the whole process. And you already know what neurotransmitter is used in that synapse, don't you? Always going to be acetylcholine, and it's always going to be excitatory. Good? So it's about the simplest anatomical route that you can have. So let's look at this image. I'm going to post this for you today because it's better than the ones in your textbook. I kind of love it. It's got like everything relatively simple. So notice what we already know from this. So this entire thing is somatic right here, somatic nervous system and autonomic. So collectively, the whole image shows the what nervous system? If it's somatic and autonomic, it collectively shows the motor or efferent nervous system. Does that make sense? Okay, you with me? Okay, so this is what we just said. We said that for the somatic nervous system, the neuron is in the central nervous system, right? Okay, and the axon is the only thing that goes through the peripheral nervous system. It releases acetylcholine. The effector is the skeletal muscle, and the effect is always excitation or stimulation, right? And the only other thing to tell you is that this is heavily myelinated because that needs to go very quickly, okay? So what we're going to do today is we're going to take this as our starting point, and then we're going to contrast it so we can learn how the autonomic nervous system is different than the somatic nervous system. Is that okay? Okay, all right. So go back to your notes. So the somatic motor neurons control skeletal muscle, or the other way to say that is skeletal muscle is the effector for the somatic motor division. The control is usually voluntary just because ultimately my cerebral cortex over on this side of my brain gets to decide when that neuron right down there fires, right? So generally speaking, skeletal muscle contraction is under voluntary control. It's not exclusively, but generally speaking, it's under voluntary control. And then we know that there is one route, and we end at a neuromuscular junction where we release acetylcholine. So let's contrast that with this. The autonomic division of the efferent nervous system is not controlled by the cerebral cortex. It is involuntary. It's controlled by much older portions of the brain, like the medulla oblongata and the pons and the hypothalamus. Okay? So it's called involuntary. Sometimes it's also called the visceral motor division, because guts. And the effectors for the autonomic nervous system are cardiac muscle, smooth muscle, and glands. Okay? The root. Okay, so the route from the central nervous system to the effector is more complicated, but it's not terrible. It's not 20 neurons. So here is what it is, and I'll come back to this figure. Okay, so the route from the central nervous system to the effector organs for the autonomic nervous system has 2 neurons in a row. It's a 2-neuron chain. And this first neuron, cell body is in the CNS, and axon runs to a ganglion. Remember that whenever you have a blob of neuron cell bodies outside of the central nervous system, it's called a what? It's called a ganglion. So this is a ganglion, okay? It only shows one cell, but there's a bunch in there. And then that second neuron goes from the ganglion, where its cell body is, to the effector. Okay, with that in mind, we will call this the preganglionic neuron and that the postganglionic neuron. Got it? Okay, no problem. Okay, the other thing that we have to understand is, let's go back to this figure for a second. Okay, so ignore this image right here. Okay, we'll get to that one in just a second. And look at this top one for sympathetic and the only one for parasympathetic. This can be confusing to students, so stick with me for a second. With skeletal muscle, I have one hookup, right? And I either stimulate it and excite it, or I just leave it the hell alone. But with the heart, because everybody knows where the heart is, I want you to think about the heart. The heart has 2 sets of hookups. It has what we call dual innervation. And dual innervation means that I have a hookup for the heart that can turn it up, and a separate hookup for the heart that can turn it down. Yes? Okay. So it's not like with my heart that I just stimulate it and it does something, and don't stimulate it and it doesn't. I can actively excite it. I can actively inhibit it. Okay? So this is called dual innervation. So please note that if you just look at the heart, you actually have the capacity to control the heart with the sympathetic and the parasympathetic nervous system. Okay, so let's talk briefly about the sympathetic and the parasympathetic, and we'll add more detail later. So the sympathetic and the parasympathetic nervous system are the 2 different branches of the autonomic nervous system. And you know the phrase that goes with sympathetic. What have you learned as the phrase that goes with sympathetic? Fight or flight. You use the sympathetic nervous system under fight or flight and exercise conditions, okay? And you use the parasympathetic under feed or breed, right? Or rest and digest conditions. So what I want to tell you is that what's going on with your heart right now is that, before we switch to images, so right now, your quiz is over, right? There's not a tiger in the room, okay? You don't have to poop, I hope, okay? So what is happening right now is, what is happening right now is you are actively inhibiting what's going on with your heart. You're keeping your foot on what your heart could do, right? So you are using which branch of the autonomic nervous system, sympathetic or parasympathetic? You're using your parasympathetic nervous system right now. And then a tiger walks into the room, and then what's going to happen is your parasympathetic nervous system says, I think this is a you job, and it's out. And your sympathetic nervous system turns on. Does that make sense? You have dual innervation. But what I want to show you is, everybody hates this figure, but it's not as bad as you think it is. I want to talk about the anatomic route to control your sympathetic and your parasympathetic nervous system just briefly. So let's go back to that same picture in which we're trying to control our heart. Right now, there's nothing going on particularly. And so what's going to happen is my parasympathetic nervous system is in control of my heart. But I want to talk about how you get there so you can appreciate the concept of dual innervation. Dual innervation says I have 2 sets of wiring hooked up. So what's going on with my heart right now is I am actually stimulating, controlling my heart via the parasympathetic nervous system, which comes, if you look right here, from cranial nerve number 10 to the heart. Cranial nerve number 10 is what? Vagus nerve. So the vagus nerve, right, the cranial nerve number 10, is right now going under my clavicle, into my thorax, and actually stimulating my heart and telling it chill, right? It's saying, calm down, you don't need to do anything, okay? So that's what's going on right now. You actually have 2 that are going to your heart, 2 vagus nerves. Okay, but then a tiger walks into the room. Then I have entirely different needs for my heart, right? So what happens now is, now I need to use the other innervation of my heart. And the way that I get there anatomically, you may or may not know. Tiger walks into the room. I need my sympathetic nervous system to stimulate my heart. And what happens is that comes from the thoracic region of the spinal cord. Do you see? Up to my heart. Okay? So it's a different anatomical route. It's a totally different anatomical route. So if a tiger walks in, it goes down here, up here, to my heart. Okay? And they're actually going to release two different neurotransmitters. Because if you're communicating with the same tissue, you can't cause two different effects by releasing the same neurotransmitter. Does that make sense? You can't, right? If you're communicating with one tissue with X receptors, you can't cause two different responses by releasing the same neurotransmitter. So we will get to the neurotransmitters. So this, when anybody looks at it initially, it's like, what the hell is that trying to show me? But what it's trying to show you is that these are all a set of organs, right? And the vast majority of them have dual innervation with the sympathetic and the parasympathetic nervous system. And it's pretty easy to see which one's which because the parasympathetic nervous system comes from 4 cranial nerves and then not a damn thing until you get down to the sacral region of the spinal cord. But the sympathetic nervous system comes from the thoracic region of the spinal cord and like a little bit in the lumbar. Does that make sense? Okay, so you can immediately look at a figure like that and tell which one it is. All right, so let's get to some functionality here. So why do we try to teach you this stuff in physiology? Because the terminology is difficult and it's a little bit hard for students to grasp sometimes. But you guys are going to go into things related to human health. And by virtue of controlling cardiac muscle, smooth muscle, and glands, the autonomic nervous system is going to control the vast majority of your systems that usually go bad on you and end you up in the hospital. Because with cardiovascular function, of course, it's mostly smooth muscle and heart and cardiac muscle, right? That makes sense? So you need to know how it's supposed to be wired up because there's a hell of a lot that can go wrong there. With respiratory function, now, of course, the diaphragm is controlled by the somatic nervous system, right? But the diameter of the bronchioles, that's controlled by the autonomic nervous system, right? Digestive function is just glands and smooth muscle partying. That's all it is down there. Urinary function is primarily blood pressure related and a little bit of glandular activity. And your reproductive function is everything is smooth muscle and glandular activity. Everything from erection to ejaculation is smooth muscle and glandular activity. Hormone secretion is glandular activity. So you need to know how this is supposed to be wired up so that you can understand the ways in which it will go bad on you. And it will. Okay, so there are 2 divisions of the autonomic nervous system. You got the sympathetic and the parasympathetic. And what the sympathetic and the parasympathetic generally do, and there are exceptions to this, is they work in concert to maintain homeostasis, right? But they're usually doing opposite things. For instance, if the sympathetic increases the activity of an organ, the parasympathetic will usually do the opposite, okay? So what we're going to do to get you guys an understanding and the capacity to predict with the autonomic nervous system is we are going to run through this table, general information about the autonomic nervous system. This is not meant to be a complete list of autonomic nervous system functions. What it is meant to do is to give you enough confidence so that you could predict one that wasn't on the list. Okay, so that's what we're going to work for. So you guys tell me, before we get into the autonomic nervous system any further, when I say autonomic nervous system, you should be able to tell me whether I'm talking about afferent or efferent function. Efferent function, yes. You should also be able to tell me whether I'm talking about CNS or PNS structures. PNS structures, exactly. It is mainly under control of old parts of the brain. The cerebral cortex is the newest part, and it's like intellect, math, and personality. These are like older. This control of cardiac muscle, smooth muscle, and glands is like alligator brain activity. It's hypothalamus, pons, medulla oblongata, mostly. It is involuntary in its functionality. Cerebral cortex is the only voluntary part of the brain. And automatic. And of course, there are 2 divisions, the sympathetic and the parasympathetic. Okay, so let's look at some sample activities. So in each particular row, what we're going to do is we're going to identify an organ, And then we are going to talk about the tissue that we're controlling in that organ. And there are only 3 choices with the autonomic nervous system of tissues to control. And they are what? Smooth muscle, cardiac muscle, glands. Exactly. So choose one of those every time. And then I am going to tell you what the sympathetic nervous system does. And then you are going to tell me whether that represents excitation or inhibition of that tissue. And then we'll do the same thing with the parasympathetic. But how do you get to the point that you can confidently predict this? And the way that you get to the point that you can confidently predict this is if the sympathetic nervous system is going to spend money or ATP on something, it needs to be helpful in the situation. Because Tiger walks into the room, we have a finite amount of oxygen and glucose and nutrients and resources, right? We have a finite amount of blood. So a lot of people are in the habit of thinking that the sympathetic nervous system, fight or flight, turns everything on. But that wouldn't be helpful, right? You would burn up your resources really quickly. So what you really need to think of it is resource allocation. Where do I need my resources to go under this situation, right? So if you're using ATP with the sympathetic nervous system, you should be able to defend why it's useful. Yeah? Okay, so let's think about it this way. So let's start with the eye. I'm going to control the diameter of the pupil. Okay? What kind of tissue is going to do that? It's going to be smooth muscle, right? Okay. So with the sympathetic nervous system, does it make sense to you that the sympathetic nervous system would cause pupil dilation? And if so, why? Why pupil dilation? Because you can see better, exactly. Okay, so if a tiger's going to eat you, you're going to be able to see every single hair, because maybe there's one that you can pull. It'll take that tiger down. Pulling here. So in order to figure out whether that represents excitation or inhibition, I have to just show you a little bit of the anatomy of the pupil. So the pupil is a hole, right? And it's got smooth muscle fibers at the edge, but the smooth muscle fibers are set up like daisy petals, right? Think of this as the origin and that is the insertion. And now I'm going to ask you, should I stimulate this and excite it and cause contraction to get pupil dilation, or should I inhibit it and cause relaxation? Yeah, I need to excite it because it's going to pull the hole open, right? That makes sense? So, excitation. And then the parasympathetic nervous system constricts the pupil back down. Okay? And that is what? Excitation or inhibition? Inhibition, exactly. Okay, salivary gland is next. What kind of tissue? Gland, yes. He's smart. Cardiac gland. Okay, salivary gland is a gland. The sympathetic nervous system decreases secretion. And I know that should make sense to you, but put it into words. Why the hell does the sympathetic nervous system decrease salivary gland secretion? Yeah, this is basically carbohydrate digestion, right? If I just ate a cracker, and then a tiger eats me, he can digest the damn cracker for himself, right? I'm not pro-tiger at that particular moment in my life, okay? So it decreases secretion. What do you think? Excitation or inhibition? Inhibition, exactly. And parasympathetic nervous system increases secretion, and that represents excitation. What I'm hoping that you are showing yourself as you go through this table, that you will dump the idea that the sympathetic nervous system stimulates everything. Because it definitely doesn't, right? And it wouldn't be helpful. Okay, so the lungs. Specifically the bronchioles. I want to control the diameter of the bronchioles. What kind of tissue? Smooth muscle, yes. And the sympathetic nervous system causes bronchial dilation. How does that make sense? More oxygen, more air. Now, in order to understand whether that represents excitation or inhibition, we have to just look at the anatomy. The anatomy is not this. It's more like this. Here's the bronchiole, and here's how the smooth muscle is arranged around it. Okay? So if I stimulate this smooth muscle, will it dilate or constrict? It will constrict. So what do I need to do? I need to inhibit it and allow it to dilate. Does that make sense? Okay. So that represents inhibition. The parasympathetic nervous system causes bronchial constriction, and that represents excitation. Now, the only thing that's kind of confusing about that is when somebody gets asthma. So when somebody gets asthma in like a stressful situation, why the hell do their bronchioles constrict? It's not nervous, it's actually paracrine. When you get into an asthmatic situation, you'll actually use boatloads of paracrine agents, which can cause bronchial constriction. And it's kind of overriding what this is trying to do. And it seems counterintuitive, but that's actually what's going on.
Can you explain that one more time? Dilate, why is it inefficient?
Because
why
this-
why do I think it's backward?
Because look, do you see how this is arranged? If I stimulate these, what it's going to do is constrict. So what I need to do is inhibit these. It's just based on the anatomy. It's just based on the anatomy. That makes sense? Okay. Heart. What kind of tissue? Smooth. You guys are failing. It is cardiac muscle. And the sympathetic nervous system increases heart rate. I don't think you need me to explain that. Increases heart rate, more blood flow, freaking tigers, that's why. And that represents excitation. Okay? And the parasympathetic nervous system decreases heart rate, and that represents inhibition. Okay, so now these next two are a little tricky, so make sure that you listen on these. They're a little counterintuitive because you might not know this yet. Okay, so what I want to do, oh wait, stomach and intestines is not tricky. Let's do that one and then we'll do the next two. Okay, stomach and intestines, I'm going to accomplish two different things with the autonomic nervous system. I have motility and I have secretion. Motility is what tissue? Smooth muscle. And secretion is glands. So you're doing both, right? Motility and secretion. Okay, so the sympathetic nervous system decreases both motility and secretion, which represents what? Inhibition. And of course it makes sense, but put it in your own words. Why do you want to decrease motility and secretion in the digestive system with sympathetic stimulation? What?
Bathroom?
Oh yeah, you don't want to have to go to the bathroom too. That's true. That's true too. But also you just can't afford the nutrients at that moment, right? You can't like afford the ATP to cause motility and secretion in the digestive tract. And also because the tiger can digest your guts for himself, right? So that's the motility and secretion, and that represents inhibition. And then, of course, the parasympathetic nervous system increases motility and secretion. This is interesting, too. I don't know why I'm talking about poop so much today. It's going to be poop or sex or something. But it's just like how my brain works. So when you almost get into a car accident or something really, really massive happens, and you almost die, what happens is you shut down your digestive tract, but then give yourself a couple of minutes for it to turn back on, and you're going to have squirts. It comes right back on, and you're like, oh, gotta go, right? So that's because it turns off and then comes right back on. One of my students told me that she had a panic attack in the last class. When her panic attack was over, she threw up. Right? You're just like, let's turn it all on. And you're like, oh, I'm going to do everything. Both directions. Okay, so now, these guys. This is where it can get confusing, so stick with me, okay? We are talking about arteries that are supplying, arteries and arterioles that are supplying two different places. I've got the arteries and arterioles supplying my skeletal muscles, right? Okay, and then I've got arteries and arterioles that are going to my skin and my guts, my ventral body cavity guts, like abdominal pelvic cavity. Okay, so what's going to happen is that is what kind of tissue in either one of them. What kind of tissue? Smooth muscle, yeah. So I'm going to control the diameter and therefore control the blood flow through those things, yes? Okay, so before we get into the details of it, I want you guys to do a prediction. So with the artery supplying skeletal muscle, upon sympathetic stimulation, those should do what? Dilate or constrict? They should dilate to send more blood to skeletal muscles, right? Just so you know, I'm not telling you that the autonomic nervous system controls skeletal muscles. I'm just talking about sending blood to skeletal muscles, right? Okay? They should dilate. I want to send more blood to skeletal muscles. But do I want to send more blood everywhere, realizing that I've only got 5 liters of blood and there's a freaking tiger? So what I need to do is need to send more blood, primarily to skeletal muscles, heart, lungs, and brain, and everything else can just F off until you're out of the way of the tiger, right? So what's going to happen in the arteries that are going to the skin and the guts, right, is those will constrict. And since there's so much skin and a lot of guts, what's generally happening is more constriction than dilation, and it tends to raise your blood pressure. Of course, your heart rate's also beating faster too, and that will raise your blood pressure. Does that make sense? Okay, so this is counterintuitive. Let's talk about it for just a second. So arteries supplying the skeletal muscle. Let's do that one first, the second one. They have the same anatomy that we were talking about with the bronchioles. but I want them to dilate upon sympathetic stimulation. So how do I cause that? Excitation or inhibition? Inhibition. Yes, I need to inhibit and therefore allow for dilation. Okay? Now, what I have is the arteries supplying the skin and the viscera have the same anatomy, okay? But upon sympathetic stimulation, I need what to do those, to do what to those? Excite or inhibit? Inhibit. Excite, yes. Excite. Now, here comes a skull cruncher. How the hell can you have the sympathetic nervous system, which always releases the same neurotransmitters, norepinephrine, okay, cause two different impacts on smooth muscle in two different places in your arteries? How? What do you think the arteries have? So, what could they have that would be different? Same neurotransmitters, different receptors. Exactly. So what happens is if I stimulate the sympathetic nervous system, it increases dilation to skeletal muscles and causes constriction to other places with the same neurotransmitter, just two different receptors on the tissues. Does that make sense? Okay. They've got different receptors. That's how they can behave that way. Okay. Last tidbit is this, and that is the adrenal medulla. The adrenal medulla, what is it? Tissue. It's a gland, exactly. So let's go back to this figure that I kind of told you guys to ignore before. Okay, so here's the adrenal medulla right here. Oh, I forgot to tell you one important thing. And the important thing is this. Blood vessels anywhere in the body don't have parasympathetic stimulation. So it's the exception to the dual innervation thing. They don't have dual innervation. So the way that you, for instance, would cause dilation versus constriction is just changing the rate of sympathetic stimulation. That's all you do. Okay, so blood vessels nowhere in the body have dual innervation. Okay, back to the adrenal medulla. Where's your adrenal gland? Where's your adrenal gland? You know where it is. No? You know where your adrenal glands are? On your kidneys, exactly. They're on top of your kidneys. Okay, so here's your adrenal medulla. The adrenal medulla has a strange little pattern, so stick with me for a second. What the adrenal medulla does is allows you to take a stressful nervous system situation and make it hormonal. Because if I get super duper freaked out, like not just a small freak out, but like almost got killed on I-15, car spinning around, slam into the guardrail, right? So you think you're going to die. What happens then is you bring in your friends. And you bring in your friends by the sympathetic nervous system stimulates the adrenal medulla. And the adrenal medulla takes norepinephrine, or the same neurotransmitter you were using, and also some epinephrine. And instead of just dumping it as a neurotransmitter, it dumps it into the bloodstream. And when you dump it into the bloodstream, it can find more targets and it lasts longer, right? And of course, it's cheaper to dump it into the bloodstream than it is to repeatedly release it as a neurotransmitter. And it's going to find a bunch of different targets that way. So you have experienced this when you almost died and then you could not calm down your heart rate immediately, right? It takes like 30 minutes for you to calm down your heart rate. Or what about this situation in which you like are completely ready for a fight with your significant other? Like you've been planning your argument the entire drive home, right? And you have planned it all out and you're thinking about, you're like, I got this argument, I got this argument, I got 27 examples over the last 16 years. I got them all, right? And you go up to my husband, not that I've ever done such a thing, and you say, Mike, we've got to talk about this. I got something I need to talk about. You did this, I felt this way about it. I have 15 other examples of the times you did it before. And he decides to say, sweetheart, you were so right. I'm really sorry. How can I help? But I've got adrenaline pumping through my veins, right? What's going to happen? You stop fighting at that point? You're like, I don't like the way you said that. I don't believe you, right? You need to go take a walk around the block. And when you're a parent, you need to remember this too, because if your kids get into full-blown flip-out mode, and then all of a sudden you're like, behave. Like, they're not going to behave. They've got this coursing through their veins. Once it gets them dumped into the bloodstream, depending on how much you have in the bloodstream, it can take 30 minutes to clear it all out. And it's 30 minutes in which your brain is primed for a freaking tiger, right? It's like tiger prime. Like when your kids are like snot bubbling and then you're like, calm down. And you yell at them to calm down. It doesn't work at all. There's more snot bubbles. So the adrenal medulla dumps neurotransmitter-like chemicals into the bloodstream and therefore enhances and prolongs the fight or flight response. Does that make sense? Okay. So, and we will talk about this one again in the endocrine system, which we will do next. Okay, so it has no parasympathetic innervation either because it doesn't need it. You either stimulate it with the sympathetic nervous system or just leave it the hell alone. Good? Okay, so last little bit about this stuff is just to put together the neurotransmitters that are used, and it's not very hard. Okay, so we're going to go back to this figure again, okay, and we're going to use the somatic nervous system as a starting point, and we're going to compare. Okay, so the organization of the synapses and neurotransmitters and effectors. Okay, so that's what we're going to do right here. So anatomic organization of all of this. The somatic, go back one more time. Just make sure that it confirms what we said. The somatic nervous system has one neuron that stretches from the CNS all the way to the effector, only one synapse. See that? Yes? That synapse is, of course, called the neuromuscular junction. The cell body of the somatic motor neuron is in the CNS. Only the axon stretches through the PNS. It's myelinated, so it's fast. There are no ganglia on the way. See that in the figure? No ganglia on the way. And the neurotransmitters that are released from somatic motor neurons, we already know this. It is always going to be acetylcholine. The effect is always excitatory. Okay, if you hit threshold, you're going to get an action potential. And you know the mechanism of excitation. The mechanism of excitation at skeletal muscle neuromuscular junctions is generally to open what kind of channels? Sodium, and cause what kind of polarization? Depolarization. And then this synapse would be considered what ergic? Cholinergic. Okay, so now use that as a starting point to compare the other two. So let's talk about the neurotransmitters that are released from the other two. So with the autonomic nervous system, of course, you're going to have a 2-neuron chain. This is the exception, by the way, right here to the 2-neuron chain. Because these second ones, by the way, the cells inside the adrenal medulla that release epinephrine and norepinephrine, they look like neurons with the axon cut off. So they're still kind of in 2-neuron chain. They just dump it into the bloodstream instead of as a neurotransmitter. But everywhere else we have a 2-neuron chain, right, between the central nervous system and the effector. Okay, so now, neuron 1 is called the preganglionic neuron, and its cell body is in the central nervous system. Neuron 2 is called the postganglionic neuron, and its cell body is in the ganglion, right? Guys, got it? Okay. So now let's look at the neurotransmitters. The neurotransmitters at autonomic neurons. So the preganglionic neurons, look up here, of both divisions release acetylcholine. Do you see that? So now I have a rule that will simplify the whole damn thing. All of this, the entire thing, is the efferent nervous system, correct? The whole thing. Look, the entire efferent nervous system releases acetylcholine first. The whole thing. Okay? And even more importantly, when acetylcholine is released first, it is always excitatory. Okay? So what I'm saying is excitatory here. I don't care that there's not a second. It's still a first, right? It's excitatory here. What I'm not saying is that it's excitatory to the effector. I'm saying that the chain doesn't stop in the middle. Okay, so I keep the thing going. So acetylcholine excitatory here, acetylcholine excitatory here, acetylcholine excitatory there, acetylcholine excitatory there. Does that make sense? Okay, so the preganglionic neurons of both divisions, parasympathetic and sympathetic, release acetylcholine. Acetylcholine binds to nicotinic cholinergic receptors on the postganglionic neuron. It's always excitatory to the postganglionic neuron. The mechanism of excitation is exactly what you would expect it to be. The mechanism of excitation is generally to open what kind of channels? Sodium, and cause what kind of polarization? Depolarization. This synapse would be what ergic? Cholinergic. So now let's be more specific. All of these synapses, 1, 2, 3, 4, are blank cholinergic. Excitatory cholinergic, all of them. Yes? Okay. All right. So all we have to do is the postganglionic neurons now. Okay. So now, conceptually, the sympathetic and the parasympathetic nervous system can't release the same neurotransmitter and cause two different effects on the same organ. So they're going to have to release two different neurotransmitters. So the postganglionic neurons in the parasympathetic nervous system, watch, are going to release acetylcholine one more time. Okay? So what does acetylcholine do? It depends on the effector. So I want you to listen to this and try to conceptualize it for me for just a second. So listen to the super wordy question and find the simple answer in it. What is the effect of parasympathetic postganglionic acetylcholine on cardiac muscle? Excitation or inhibition? Inhibition, because the important word there is that it's parasympathetic, right? What does the parasympathetic do to cardiac muscle? It inhibits it, yes? And it does it by parasympathetic postganglionic acetylcholine, right? Does that make sense? Okay, but now ask a similar question. What is the effect of parasympathetic postganglionic acetylcholine on the smooth muscle of the stomach? Parasympathetic postganglionic acetylcholine. Excitation, because you know that the parasympathetic nervous system excites the smooth muscle of the digestive tract, right? So it's super wordy, but it's a really simple concept. That's it, okay? So last thing is this. Postganglionic neurons in the sympathetic division release norepinephrine and epinephrine, which are very similar to one another, but they sometimes interact at slightly different receptors. Okay, this synapse right here would be considered what ergic? Adrenergic, exactly. Okay, so how do we predict what's going to happen? Let's try it one more time. What is the effect of sympathetic postganglionic norepinephrine on cardiac muscle? Excitation, yeah, because sympathetic nervous system excites cardiac muscle. All I'm trying to do is get you to add in the players there, right? Okay, what is the effect of sympathetic postganglionic norepinephrine on the smooth muscle of the stomach? Inhibition, yes, because the sympathetic nervous system inhibits digestive function. So when you get to this part right here, you just have to go back to your resource allocation thinking. To me, that is not memorization, because I can throw you one that I haven't told you before, but I bet you can get it. You ready? Okay. What is the effect of, think about it, parasympathetic postganglionic acetylcholine on on salivary gland secretion? Excitation. What is the effect of sympathetic, stick with me, sympathetic postganglionic norepinephrine on the smooth muscle of the uterus? Inhibition. Do you see? It's in there. You just need to practice with the terminology, and then you'll be fine. Okay? Okay. Now, here's one for you. What is the effect of parasympathetic preganglionic acetylcholine? Excitation. I don't even have to say the rest of the sentence, do I? Parasympathetic preganglionic acetylcholine, excitation, right? So we know, do you see that the entire thing simplifies so that you can see only one place do you use anything other than acetylcholine, and that's where? Postganglionic sympathetic, that's the only place you use anything other than acetylcholine, right? And of course, you've already got the excitation or inhibition memorized with all of those first ones, right? That makes sense? So the effects of a postganglionic neurotransmitter can be excitatory or inhibitory to any tissue, because it doesn't really depend on the neurotransmitter, it depends on the what? Receptor. It depends on the receptor that it interacts with. Is that okay? Yeah? Okay, so that is autonomic nervous system. Let me tell you, I'm not going to test over this, but I'll tell you something interesting just to make sure that you can sort of bring this home for a second, because we've got 4 minutes. Okay, ready? Let's talk about lie detector tests. Let's talk about polygraph tests. Okay? What a polygraph test has to do with this story? Well, it's kind of an interesting thing. With the polygraph test, what they're doing is they are using external measurements to see what your sympathetic nervous system is doing. So I've never had a polygraph. I swear I will volunteer for one anytime. If any of you guys knows a cop who will give me a polygraph.
They're ugly.
Huh? They are?
I was in one for 5.5 hours.
5.5 hours?
They told me I was a terrorist or a spy or just a good guy trying to beat the exam. So.
You were a terrorist or a spy? Oh, you're a good liar, huh?
Wow.
I wonder if I'm a good liar. I suspect I'm a really bad liar. I don't know though. Maybe I'm lying to myself and I'm really good at it. Okay, so what they do is there's multiple different ways depending on how expensive a polygraph test you want to do. They can measure heart rate, of course, right? They can measure increased conductivity of the skin because the sympathetic nervous system tends to increase your body temperature and therefore you start to sweat, right? It doesn't even have to be major. The fancier ones can measure pupil dilation, right? And they can measure increased respiration, which isn't directly related to the sympathetic nervous system, but tends to follow along with the sympathetic nervous system. So what they do is they ask you a series of baseline questions. Maybe you can tell us. They ask you a series of baseline questions like what you had for breakfast and what's your mother's maiden name and all that stuff to get your baseline level of sympathetic stimulation, because a lot of times you're a little stressed out because you're having a freaking lie detector test, right? But then they start throwing questions at you, and the questions at you will get closer and closer to the things that they're interested in. So if they think you killed somebody and cut her up and put her in your trunk, they're going to ask you if you were hanging out with her last Thursday or if you saw her last Thursday, right? So they're going to ask you. And what happens is when you lie, because of our fear of the social consequences of our behavior, your sympathetic nervous system kicks up. So it gives you a little spike whenever you lie because you know that the possibility exists that you could actually get caught or some kind of social ostracism. And then after they get that, then they'll ask you, okay, well, did you buy the gun, right? Do you own a gun? Did you cut her up? Did you stick her in your trunk, right? Is she in your trunk right now? And they continue to ask these questions, interspersed with questions that are completely baseline to see whether you're just really getting stressed out. And when you spike on all of the things that they were interested in, the only thing that really can happen is that they can't exclude you as a suspect. But in the United States, we actually cannot include lie detector tests or polygraph tests in any criminal proceeding at all. So they're inadmissible in criminal proceedings. So if you know that you didn't do it, you can go ahead and volunteer for a lie detector test. Because if they asked you, you were already a suspect for the lie detector test. So, so that's how those work. Now, what about spies? What about the CIA agent or the yogi who can take a lie detector test and not have any sympathetic stimulation spike? Well, what can happen is you can train yourself to those things, right? It's just like you do a biofeedback, right? So when you're doing biofeedback, if you can feel what's happening, then you can override it with your cerebral cortex, and your cerebral cortex tells your hypothalamus to shut the hell up. And you do it effectively enough so that you can feel it when it's actually happening, and then your results become teeny tiny spikes and not big enough to actually detect at all. So you can start to get to be a really, really effective, really good liar when that occurs. So CIA operatives and spies and yogis and those kinds of things, those people who can like not have what? Or the other people who don't have lie detector responses are sociopaths. Seriously, so if you do not have any particular fear of social approbation, like other people literally don't mean a damn thing to you, then you can lie at will. And it doesn't make any difference. Ted Bundy, do you know who Ted Bundy is? Serial killer. He passed like 3 lie detector tests before they ever caught him. Because he was like, sure, I'll volunteer for a lie detector test because I literally don't care and I would kill you just as easily. So that's how they work. But I've never taken one. I'm curious. 5-hour lie detector test.
It was crazy. I was applying for the Secret Service.
Oh, you were.
For a year and a half. I mean, I take tests, interviews.
You could do that.
Oh my God! I have so many skeletons in my closet.
They had people.
Hell.
They had people looking through a stained glass, like a smoke glass mirror at me. Just
Is that why you take the job?
well, they basically, after about 5 hours, I was so frustrated. I was,
You're like, I don't want to do this
I
job.
just basically broke down and cried. So it was ugly.
Oh. So they actually talk to the person.
They asked me like baseline questions were, have you ever done anything in your adult life that you're ashamed of? Whatever. You know, I went on for a minute. Do you think that's honestly what we're talking about? Probably not. Then after the first 30 minutes, they said, get up and leave. And come back when you tell me the truth. Well, what? And I was sitting on this little mouse pad kind of thing. Thinking, they said, you know, they said my sphincter muscle was going crazy.
Oh, they were testing your sphincter muscle.
Well, they said,
Oh my God, they got fancy ones.
they said, this is something they teach you how to, you know, do to throw off the system on the internet. And I'm like, I haven't been on the internet looking to, I have nothing to hide.
Really?
They said, you have had illegal contact we asked you if you had ever had illegal contact with a foreign government. You were off the charts. Your sphincter muscle was going crazy.
I didn't know they tested sphincter muscle. So they have the expensive ones.
Oh, yeah,
They love to recruit in Utah for CIA and FBI.
Mississippi. I had to move back to Mississippi.
Oh, because of all of our squeaky clean Mormon boys with the right haircut and they already have the cheap suit. So
Dropping the test off over there tomorrow, Wednesday.
I'll send it, email. Mm-hmm.
Question for you.
What is a good way to study?
I'm going to teach you one if you want to come for
I can't today because I'm nannying my brother.
I